Hello, and welcome to an abbreviated overview of the Lac Operon. Here we have a petri dish full of happy little bacteria colonies. It's the only thing we can realistically see on the macroscopic scale. But that doesn't stop us from speculating, from knowing exactly what goes on at the cellular level. So let's get in a little closer and see what that's like. Here's a nice, suitable cell. For clarity, we'll look at a very stylized cross-section of this one, and just for us, it'll be so kind as to not divide. Here also is a visualization of cell density with time. A popular metric is optical density, in this case, the absorbance of light with a wavelength of 595 nanometers. Oh look, there seems to be a lot of lactose around. Now, the cell is perfectly capable of processing lactose, but it doesn't make sense to do that when there isn't any. And so the Lac Operon controls when the cell builds the machinery to use lactose, and when it doesn't. Now, for instance, is a good time to be making these transmembrane proteins, which we'll call lactose permease, to allow for its intake. This is known as inducible expression. External lactose will induce the cell to reconfigure itself to use it. After a brief lag period, the cell can very efficiently use the lactose in its environment and enter a log period of exponential growth, at least until we reach carrying capacity. Of course, when the lactose is gone, transcribing and translating the necessary proteins amounts to a complete waste of energy. So lactose permease levels respond in kind as the operon shuts down new production. Cell structures don't have infinite lifespans, so simply not replacing something will make it less common. Well, that's about all we can get from way out here. On the surface of the cell, as you can see right now, there are many proteins of all shapes and sizes for all sorts of jobs, none of which we'll be talking about today. What we're really interested in is the inside of the cell. So, how do we... here's a residual lactose molecule we can hitch a ride on. I'm sure it won't mind. Here we meet beta-galactosidase for the first time. Lactose is classified as a beta-galactoside, so beta-galactosidase, or lactase, processes lactose. There are two things it can do. The first is to facilitate the splitting of lactose into glucose and galactose. Both are monosaccharides that can then be further metabolized. It also turns some small amount of lactose into an isomer we call allolactose. This will become important later. But first, let's take a look at how our proteins are made. As it turns out, lactose permease and beta-galactosidase are found on the same set of regulated genes. This is what's known as an operon. An operon is transcribed into a polycistronic mRNA, which means, actually, you know what, let's find out. So here we have the LAC-Z, LAC-Y, and LAC-A genes. Here comes RNA polymerase to transcribe the DNA, okay good, and not one, not two, but three ribosomes translating the mRNA all at once. And no, we really don't know what the LAC-A protein actually does. It's called transacetylase if you must know. Be free, good proteins. The mark of an operon is its control under a single promoter. So let's take a look at that promoter region. Ordinarily, it would be next to the start of the gene, but in this case, there's an operator in between the promoter and LAC-Z, to which the repressor binds. Also keep this part in mind. We'll call it the cap binding site, because it's the site that binds the metabolite activator protein. It's also called CAMP receptor protein and is activated by, you guessed it, CAMP. I'm going to call it CAP from here on out just because it's easier to say. The last thing to note is that there's a separate gene quite a ways upstream, LAC-I, that codes for the repressor we mentioned earlier. By the way, say hi to all the other proteins floating around here. Hi, proteins. The repressor is a trans element, as opposed to being a cis element, which we'll talk about later. A trans element can go anywhere in the cell, and indeed this particular repressor will just go and chill out with all the other ones out there. The LAC-I gene, which is constitutively expressed to maintain a steady amount of protein at all times, is sometimes not considered part of the operon. but it does have a noteworthy operator site on it as well. The actual action of this repressor, a tetramer, is to bind to these two operator sites, rendering the promoter inaccessible. But for simplicity, let's represent that with a simple wall that blocks the RNA polymerase from binding to the DNA. Remember our good friend allolactose? Well, when the repressor inevitably comes off, as it does from time to time, that's how allolactose becomes relevant in the first place, it binds to a special site and prevents it from reattaching to the operator. Congratulations, the operon is now open for business. But as it stands, transcription still has a rough time getting started. Now, to truly kick off the party, we need the binding of cap, which greatly increases the affinity of RNA polymerase to the promoter. Alright, now that cap's comfortably on the promoter, let's see what happens to the rest of the oh no, what have we done? What we've done is increase the expression of these genes to the max, because sometimes this level of transcription is very useful to have. We'll talk about that momentarily, but first let's take a detailed look at how CAMP, and therefore CAP, is regulated. When the cell takes in glucose, it is immediately phosphorylated by 2A. This phosphate group comes from a long chain of members, namely from PEP to E1 to HPR to 2A, and so it's like a conveyor belt that finally takes the phosphate to the incoming glucose. This is useful because diffusion, which is how glucose crosses the membrane, stops naturally when concentrations inside and outside the cell are equal. But there's a loophole here. When glucose is phosphorylated, it doesn't count anymore, so inside the cell concentrations remain effectively at zero no matter how much the cell takes in. When there's no glucose around, however, 2A stays phosphorylated long enough to run into adenylyl cyclase, which it's also responsible for activating. Adenylyl cyclase is then able to turn ATP into CAMP. Thus, low glucose levels result in more CAMP and more activated caps. The inverse is also true. High glucose levels equal inactive caps. So let's review. With only glucose around, the Lac Operon remains repressed, and cap is inhibited as well. The operon is only minimally transcribed, and expression of lactose proteins is low. Glucose transporters, on the other hand, undergo high expression. When there is only lactose, the repressor is off and cap is on, and the operon becomes very active. The production of lactose proteins then increases while glucose transporters are neglected. When the cell is put in a medium with glucose and lactose, things get a bit more complicated. The repressor is off, but since the activator is 2, Lac Operon expression remains low, and we get more glucose transporters as before. But once glucose runs out, cap is activated, and it kickstarts the expression of the operon as the cell shifts to lactose as its energy source of choice. This takes a while, and cell growth is arrested in the meantime. But eventually, it grows just as robustly as before, despite having to adapt to new conditions. All of this is with a single Lac Operon. And what's better than one Lac Operon? 2 Lac Operons. Okay, first, let's talk about how things can go horribly wrong. There are the 3 lactose proteins. Each one can mutate, which will probably leave it non-functional. That's straightforward. But what if we mutate the other things? Here, let's set up a code. If the cell can still express the operon on a lactose medium, I'll flash green. If it can't, red. And let's work backwards. Now, if the operator mutates, the operon is still intact, but can't be turned off. If the promoter fails, that's a strong no. If the cap binding site fails, the cell can never transcribe at a useful rate, so still no. These 3 are cis elements, which means they can only affect the particular operon they're a part of. The trans element Lac-I, though, has no such limits. This protein can be mutated at 2 sites. The first is the operator binding site for a non-functional repressor, which is similar to having a broken operator. The other is the allosteric binding site, which makes it unresponsive to allolactose. And that's devastating, since it shuts down the operon under all conditions. You could play this game with different combinations of mutations on each of our 2 operons, but here's one situation that's somewhat illustrative. This operon makes a normal repressor, and this one makes a super repressor. Everything else is normal. Will this operon make any lactose proteins? How about this one? I'll give you a chance to pause and think about it. Okay, time's up. So it turns out the answer is no. And here's why. When these repressors are made, they might have a good chance of sticking onto their own operons at first, but they can migrate elsewhere given enough time, and trust me, we are made of time. Allolactose binds with the normal repressors and deactivates them. But these super repressors, they don't care. They'll latch onto both operators, and just about nothing is going to get them to stay away. So why does this matter to us? Well, in short, when everything's working at least, the Lac Operon fine-tunes the cell's activities so that it can optimize the use of available energy resources. And that means the cell colonies can grow as quickly and efficiently as possible. And that makes for very happy biologists.